For vocabulary, see Glossary of gene expression terms. For a non-technical introduction to the topic, see Introduction to Genetics. Genes are expressed by being transcribed into RNA, and this transcript may then be translated into protein. Gene express. ION is the process by which information from a gene is used in the synthesis of a functional gene product. These products are often proteins, but in non-protein coding genes such as transfer RNA, tRNA, or small nuclear RNA, snRNA, genes, the product is a functional RNA. The process of gene expression is used by all known life eukaryotes, including multicellular organisms, prokaryotes, bacteria and archaea, and utilized by viruses to generate the macromolecular machinery for life. Several steps and the gene expression process may be modulated, including the transcription, RNA splicing, translation, and post-translational modification of a protein. Gene regulation gives the cell control over structure and function, and is the basis for cellular differentiation, morphogenesis, and the versatility and adaptability of any organism. Also, it depends on the state of activator RNA. Gene regulation may also serve as a substrate for evolutionary change, since control of the timing, location, and amount of gene expression can have a profound effect on the functions, actions, of the gene in a cell or in a multicellular organism. In genetics, gene expression is the most fundamental level at which the genotype gives rise to the phenotype, i.e. OBSE. RBA will trait. The genetic code stored in DNA is interpreted by gene expression, and the properties of the expression give rise to the organism's phenotype. Such phenotypes are often expressed by the synthesis of proteins that control the organism's shape, or that act as enzymes catalyzing specific metabolic pathways characterizing the organism. Regulation of gene expression is thus critical to an organism's development. Mechanism. The process of transcription is carried out by RNA polymerase, RNAP, which uses DNA, black, as a template and produces RNA, blue. Main article, Transcription, Genetics. A gene is a stretch of DNA that encodes information. Genomic DNA consists of two anti-parallel and reverse complementary strands, each having 5' nd 3' ends. With respect to a gene, the two strands may be labeled the template strand, which serves as a blueprint for the production of an RNA transcript, and the coding strand, which includes the DNA version of the transcript sequence. Perhaps surprisingly, the coding strand is not physically involved in the coding process because it is the template strand that is read during transcription. The production of the RNA copy of the DNA is called transcription, and is performed in the nucleus by RNA polymerase, which adds one RNA nucleotide at a time to a growing RNA strand as per the complementarity law of the bases. This RNA is complementary to the template 3' 5' DNA strand, which is itself complementary to the coding 5' 3' DNA strand. Therefore, the resulting 5' 3' RNA strand is identical to the coding DNA strand, with the exception that the mines T are replaced with uracils U in the RNA. A coding DNA strand reading, ATG, is indirectly transcribed through the non-coding strand as August. In RNA, in prokaryotes, transcription is carried out by a single type of RNA polymerase, which needs a DNA sequence called a Pribnow box as well as a sigma factor to start transcription. In eukaryotes, transcription is are formed by three types of RNA polymerases, each of which needs a special DNA sequence called the promoter and a set of DNA-binding proteins, transcription factors, to initiate the process. RNA polymerase I is responsible for transcription of ribosomal rRNA genes. RNA polymerase II, whole 2, transcribes all protein-coding genes but also some non-coding RNAs, e.g., snRNAs, SNORTAs or long non-coding RNAs. Whole 2 includes a C-terminal domain, CTD, that is rich in serine residues. When these residues are phosphorylated, the CTD binds to various protein factors that promote transcript maturation and modification. RNA polymerase III transcribes 5srRNA, transfer RNA, tRNA genes, and some small non-coding RNAs, e.g., 7sk. Transcription ends when the polymerase encounters a sequence called the terminator. Main article, Post-transcriptional modification. While transcription of prokaryotic protein-coding genes creates messenger RNA, mRNA, that is ready for translation into protein, transcript Ion of eukaryotic genes leaves a primary transcript of RNA pre-mRNA, which first has to undergo a series of modifications to become a mature mRNA. These include 5' capping, which is a set of enzymatic reactions that add 7-methylguanosine, M7G, to the 5' end of pre-mRNA and thus protect the RNA from degradation by exonucleases. The M7G cap is then bound by cap-binding complex heterodimer, CBC20, CBC80, which aids in mRNA export to cytoplasm and also protect the RNA from decapping, another modificate. Ion is 3' cleavage and polyadenylation. They occur if polyadenylation signal sequence 5-AAUAAA3 is present in pre-mRNA, which is usually between protein coding sequence and terminator. The pre-mRNA is first cleaved and then a series of approximately 208 9s A are added to form poly-A tail, which protects the RNA from degradation. Poly-A tail is bound by multiple poly-A binding proteins PABP, necessary for mRNA export and translation ray initiation, simple illustration of exons and introns in P. Ray mRNA and the formation of mature mRNA by splicing. UTRs are non-coding parts of exons at the ends of the mRNA. A very important modification of eukaryotic pre-mRNA is RNA splicing. The majority of eukaryotic pre-mRNAs consist of alternating segments called exons and introns. During the process of splicing, an RNA protein catalytical complex known as spliceosome catalyzes two transesterification reactions, which remove an intron and release it in form of lariat structure, and then splice I bring exons together. In certain cases, some introns or exons can be either removed or retained in mature mRNA. This so-called alternative splicing creates series of different transcripts originating from a single gene. Because these transcripts can be potentially translated into different proteins, splicing extends the complexity of eukaryotic gene expression. Extensive RNA processing may be an evolutionary advantage made possible by the nucleus of eukaryotes. In prokaryotes, transcription and translation happen together, whilst in eukaryotes, the nuclear membrane separates the two processes, giving time for RNA processing to occur. Main articles: tRNA maturation, rRNA maturation, and mRNA maturation in most organisms non-coding genes. ncRNA are transcribed as precursors that undergo further processing. In the case of ribosomal RNAs, rRNA, they are often transcribed as a pre-rRNA that contains one or more rRNAs. The pre-rRNA is cleaved and modified to O-methylation and pseudoridine formation at specific sites by approximately 150 different small nucleolus-restricted RNA species called SNORNAs. SNORNAs associate with proteins, forming SNORN, while SNORNA part base a pair with the target RNA and thus position the modificate on at a precise site. The protein part performs the catalytical reaction. In eukaryotes, in particular ASNORNP called RNase, MRP cleaves the 45S pre-rRNA into the 28S, 5.8S, and 18S rRNAs. The rRNA and RNA processing factors form large aggregates Ca. LLED the nucleolus. In the case of transfer RNA tRNA, for example, the 5' sequence is removed by RNA-SCP, whereas the 3' end is removed by the ternase Z enzyme and the non-templated 3' CCA tail is added by a nucleotidal transferase. In the case of microRNA, mRNAs are first transcribed as primary transcripts or PRI mRNA with a cap and poly-A tail and processed to short 70 nucleotide stem loop structures known as pre-mRNA in the cell nucleus by the enzymes DRASA and PASHA. After bind G exported, it is then processed to mature mRNAs in the cytoplasm by interaction with the endonuclease dicer, which also initiates the formation of the RNA-induced silencing complex RISC composed of the argonaute protein, even snRNAs and SNORNAs T. Hem cells undergo series of modification before they become part of functional RNP complex. This is done either in the nucleoplasm or in the specialized compartments called Cajal bodies. Their bases are methylated or pseudoridylated by a group of small Cajal body-specific RNAs, scarnas, which are structurally similar to SNORNAs. In eukaryotes, most mature RNA must be exported to the cytoplasm from the nucleus. While some RNAs function in the nucleus, many RNAs are transported through the nucleus pores and into the cytosol. Notably, this includes all RNA types involved in protein synthesis. In some cases, RNAs are additionally transported to a specific part of the cytoplasm, such as a synapse. They are then towed by motor proteins that bind through linker proteins to specific sequences called zip codes on the RNA. During the translation, tRNA charged with amino acid enters the ribosome and aligns with the correct mRNA triplet. Ribosome then adds amino acid to growing protein chain, N article, translation, genetics. For some RNA, non-coding RNA, the mature RNA is the final gene product. In the case of messenger RNA, mRNA, the RNA is an information carrier coding for the synthesis of one or more proteins, mRNA carrying a single PR. Otine sequence, common in eukaryotes, is monocistronic, whilst mRNA carrying multiple protein sequences, common in prokaryotes, is known as polycistronic. Every mRNA consists of 3 parts, a 5' untranslated region, 5 UTR, a protein coding region or open reading frame, or, and a 3' untranslated region, 3 UTR. The coding region carries information for protein synthesis encoded by the genetic code to form triplets. Each triplet of nucleotides of the coding region is called a codon and correspond DS to a binding site complementary to an anticodon triplet and transfer RNA. Transfer RNAs with the same anticodon sequence always carry an identical type of amino acid. Amino acids are then chained together by the ribosome according to the order of T. Riplets in the coding region. The ribosome helps transfer RNA to bind to messenger RNA and takes the amino acid from each transfer RNA and makes a structure-less protein out of it. Each mRNA molecule is translated into many protein molecules, on average. Age approximately 2,800 in mammals. In prokaryotes, translation generally occurs at the point of transcription, co-transcriptionally, often using a messenger RNA that is still in the process of being created. In eukaryotes, translation can occur in a variety of regions of the cell depending on where the protein being written is supposed to be. Major locations are the cytoplasm for soluble cytoplasmic proteins and the membrane of the endoplasmic reticulum for proteins that are for export from the cell or insertee on into a cell membrane. Proteins that are supposed to be expressed at the endoplasmic reticulum are recognized partway through the translation process. This is governed by the signal recognition particle, a protein that binds to the ribosome and ECTS it to the endoplasmic reticulum when it finds a signal peptide on the growing, nascent, amino acid chain. Translation is the communication of the meaning of a source language text by means of an equivalent target language text. Main article, prot. In folding protein before, left, and after, right folding, the polypeptide folds into its characteristic and functional 3-dimensional structure from a random coil. Each protein exists as an unfolded polypeptide or random coil when translated from a sequence of mRNA into a linear chain of amino acids. This polypeptide lacks any developed 3-dimensional structure, the left-hand side of the neighboring figure. Amino acids interact with each other to produce a well-defined 3-dimensional structure, the folded protein, the right-hand side of the figure, known as the native state. The resulting 3-dimensional structure is determined by the amino acid sequence, Anfinson's dogma. The correct 3-dimensional structure is essential to function. Although some parts of functional proteins may remain unfolded, failure to fold into the intended shape usually produces inactive proteins with different properties, including toxic prions. Several neurodegenerative and other diseases are believed to result from the accumulation of misfolded proteins. Many allergies are caused by the folding of the proteins, for the immune system does not produce antibodies for certain protein structures. Enzymes called chaperones assist the newly formed protein to attain, fold into, the 3-dimensional structure it needs to function. Similarly, RNA chaperones help RNAs attain their functional shapes. Assisting protein folding is one of the main roles of the endoplasmic reticulum in eukaryotes, secretor. Why proteins in eukaryotes or prokaryotes must be translocated to enter the secretory pathway. Newly synthesized proteins are directed to the eukaryotic Sec61 or prokaryotic Sec-YEG translocation channel by signal peptides. The efficiency of protein secretion in eukaryotes is very dependent on the signal peptide which has been used. Many proteins are destined for other parts of the cell than the cytosol, and a wide range of signaling sequences or signal peptides are used to direct proteins to work they are supposed to be. In prokaryotes, this is normally a simple process due to limited compartmentalization of the cell. However, in eukaryotes there is a great variety of different targeting processes to ensure the protein arrives at the correct organelle. Not all proteins remain within the cell and many are exported, for example, digestive enzymes, hormones and extracellular matrix proteins. In eukaryotes the export pathway is well developed and the main mechanism for the export of these proteins is translocation to the endoplasmic reticulum followed by transport via the Golgi apparatus. Transcription. The process of transcription is carried out by